Hi, I'm Meredith Blackwell,
And I'm Don Pfister.
and this is,
Marc Cubeta.
and we are here at the Mycological Society of America meeting, our annual meeting. It's being held on the campus of the University of Florida, and we're in the Hilton Hotel on the campus, and the date is 10th of July, 2022. So, here's an ex-president, and what else is on that? May I see your name tag?
Yeah.
Okay, past president and MSA fellow. So he's a decorated mycologist here within the Mycological Society of America. And he's really a very recent past president. So why don't you tell us something about yourself, starting at the beginning?
Okay, so we'll just go back to the beginning. I was born in Charleston, South Carolina, and my mother and my father were actually in the military, in the Air Force. And so I was a military baby, and then after they had served their terms in Charleston, then they moved back to Pennsylvania, central Pennsylvania.
You didn't sound real Southern.
Well, I get this all the time. People always ask me, if I'm Southern, and they recognize that my accent's from the northern part, particularly Pennsylvania. They always say, "Are you from Philadelphia?" I'm like, "No, I'm from central Pennsylvania." But yeah, so I always tell them that I'm Southern by birth, right? But northern where I was raised. Pretty much, yeah. So yeah, I do have, I guess, some Southern blood because I was born here, but I didn't spend much of my time here. So I spent most of my time in central Pennsylvania, and also my parents moved back to Delaware, where I basically went to grade school and high school, and I also went to college there at the University of Delaware because I was the first person in my family, the whole entire family basically, to go to college.
So what were your parents doing in the military? Did they have a special, both of them were in?
Yeah, my dad actually worked on planes. So he's was a mechanic, and he fixed plane engines, and that's why he can't hear today, because he basically was around a lot of loud engines. There wasn't a lot of protective equipment, I guess, back then. And then my mother was also involved in flights and things like that, trying to organize flights. So.
Interesting. Yeah.
So your college was where?
I went initially to the University of Delaware, which is in Newark, Delaware. I got a bachelor's degree there. And
in biology?
Yeah, I didn't really know what I was doing. I went to school. I started out as my uncle actually worked for the FBI, and he was in law enforcement. And he was always trying to solve crimes, and I was like, well, that would be really interesting to try to do that, right? So I was way before all these CSI shows, I was thinking about, I didn't want to be one of the medical examiners, but I wanted to be someone that maybe could, so I was always interested in solving things. But then as time went on, I just said, nah, that's really not what I do. So I think it was the second semester when I was a sophomore, I took a soils class. I believe I took a plant science, plant pathology class, and also a microbiology class. Mm-hmm. So those were the three classes, and it finally clicked in my brain. I was like, oh yeah, I could work with organisms that live in soil that are associated with plants. And I kept asking the teacher in microbiology, why aren't we looking at microbes associated with plants? And if you go back to my mother, when I was a little child, I was always out in the yard, out in the woods, playing with stuff. So she figured that I'm going to be doing something with soil, or plants. So it was probably, early brought to me sometime in my childhood. So,
dirty kid.
Yeah, I was a dirty kid. She was always trying to clean me up. So yeah, so at the University of Delaware, I took those courses, and then I started taking more soils courses, and I actually had an opportunity to work in the soils lab, soil chemistry lab of Dr. Sparks, and I also worked with a botanist there because at that time they had opportunities to work in the labs and also teach, and I didn't know any of this existed. And I went to college on what's called a BEOG fellowship, so basically they're called Pell Grants now. Okay. So they're basically for people that don't have a lot of money, they want to go to college. But if you get one of those awards, then you have to every semester work. So I was working a lot of times. I'd work in the cafeteria. Yeah. I used to teach kids how to play ice hockey and things like that. So as I got further advanced, I started thinking there were opportunities to teach. And so they provided me with an opportunity to teach. And it was kind of unusual at that time because, an undergraduate student actually involved in a lab. I was actually teaching a botany lab, which is, to me, very unusual now that I know that most graduate students do that. So I had an opportunity to do that. And there were two great advisors that basically, his name was Tom Pizzolato, he was a botanist, and Donald Sparks, who's now in the National Academy of Science. He's a soil chemist. So little did I know, I was working with some really impressive people. And I was just glad to have an opportunity to do anything. And I really love teaching, and doing research. So I think that kind of jettisoned me to my sort of next, I guess, thing that I was going to do in my life. But they prepared me really well for graduate school because they kept saying to me, you might want to go to graduate school someday. And I'm like, I don't know. Because I didn't know about graduate school. And but no, you need to take this course. You should take this physical chemistry. So all my classmates were taking all these other classes, and I was telling them I was taking all these really hard courses. And they're like, why are you taking those courses? I said, my advisor said I had to take these. But when I went to graduate school, then it really helped me, because they gave me an entrance exam in Illinois, and I had to pass this entrance exam. And then they were asking me questions about viruses, and I didn't really know about viruses, so I told them I was just going to take the course,
So you went from Delaware to a
University of Illinois.
master's?
Yeah, so I got a master's degree at the University of Illinois, and at that time I was working on a project where I was looking at the fungi that are associated with soybean seeds, and I was also looking at sort of the bacteria and trying to figure out, could I see interactions between bacteria and fungi. And at that time, Dr. Gottlieb was there. I don't know if you know him. He was actually an old professor that had worked with Selman Waxman, at Rutgers University. They were the initial discoverers of antibiotics, And so, just amazing, this older professor, he would actually talk to me. I was and he would tell me these stories about how they discovered the antibiotics. So I got really excited about that. And then I took a course by Dr. Crane. I don't know if you know Lee Crane. Yeah, so we took his mycology course, and something just clicked in me there. I was just thinking, this is what I got to do. And then also at that time, one of my close colleagues, his name's John McPartland. He's a medical doctor now in Vermont, but he would actually, for his projects, he'd be describing fungi. And I didn't know much about taxonomy, so I was trying to learn some of that from him. But as he was writing sort of his papers, we'd have to have Latin descriptions. So we'd actually go over to Dr. Rogers' house. Don Rogers? Yeah, and Rogers would actually do the Latin description. So I got a chance to interact with him when I was there as well. And lo and behold, Rogers, he described all this, ceratobasidium, right?
Yeah,
and that's what I eventually ended up working with, which is amazing, right?
Yeah.
He's this,
Came around.
Yeah, so something happened to me, I guess, when I interact with Dr. Rogers, that I ended up working with Rhizoctonia-like fungi, and But then after that, I decided I need a break. I didn't, I was they wanted me to continue on for a PhD, and I had a lot of opportunities, but I just decided that I needed to step back for a while. And then I went to California, and I worked for, it was a company called Upjohn at that time. Oh, yeah. They actually made vitamins, and their famous product is minoxidil, right? Because that's the one for growing the hair. Yep. They used to call it Regain, but the FDA said they have to call it Rogaine instead of Regain because it was a little bit misleading. So at that time, they had an agricultural sort of division, and where they would actually do plant genetics. And of course, plants and seeds, they get bacteria and fungi associated with them. So that was my job. I basically tried to identify bacteria and fungi that were in seeds, but I also worked a lot with plant breeders on trying to develop new lines that would be resistant to disease. And so that was sort of my step way back from, I guess, academics for a while. So this is what I tell the students all the time, that it's okay. You don't have to go through, you know, get a bachelor's, get a master's, get a PhD. Now it's a postdoc. But yeah, you can take time off. And I really see that students that do that, they really have a different perspective on, you know, graduate school, because they've taken time to think about what they really want to do with their lives. So I see those students as, a lot of times, they're more engaged students when they start to, you know, start their programs. So.
Yeah. You did that for a while, but you obviously went on.
Yeah, so then after 4 or 5 years, I don't know, I guess you could say I was at the end of my career path because there wasn't much more to go there. And of course, they wanted me to stay there. There were other opportunities to move to other companies, and I just decided that I needed to really go back to school. So I applied to go to graduate school at UC Davis, University of Wisconsin, Cornell, and North Carolina State University. And I was accepted at all those institutions. And I had to decide where I was going to go. And so I was leaning towards the east because my father and my mother kept asking me, am I going, well, first you went to Illinois. Now you went to California. Are you going to like be going to Hawaii, Japan next? Yeah, so they were like, are you going to, you know, continue to go westward? Or are you going to come back? And so I figured that I would really like to try to get back east. And it really came down to Wisconsin. Cornell didn't have any assistantships, even though later they actually came back and offered me an assistantship. But by that time, I'd already accepted. But and then I just decided, well, you know, North Carolina State University is really where I want to go. So again, continued on the plant pathology sort of theme there. But that's where I met Redis, right? Because during my degree, I saw Redis give a talk at a meeting. I think it was 1988, and I just thought it was so cool. I was like, oh my God, you know, he, you know, is doing all this stuff on the fungus I'm working on. And I asked my advisor, you know, hey, is it okay for me to go work in this lab? And of course, my advisor at that time was Eddie Ashandi, and he was a student of J.C. Walker, who was a very famous plant pathologist. And so he was always encouraging me to do things, reaching out beyond what I was doing. And then he said to me, well, yeah, that's fine with me. And then, but after I found out later, almost all my committee members didn't want me to do this. They were trying to prevent me from doing it because they thought that I wasn't going to do anything. Oh, he's just going to go over there. But it turned out to be probably one of the best things I ever did in my entire life. And so I owe Redis a, you know, big debt of gratitude for, you know, all his mentoring over the years. And now I know his family. And I was his second postdoc that he had. So it was back in the days, I was telling Meredith that when they were just starting out with PCR, right? And I believe we have one of the first papers on a fungus, at least on a plant pathogenic fungus, using PCR. But that's when John Taylor and Brunson, they were, you know, developing all these primers. And we had access to these primers that they were like, you know, can you test these for us? And we're like, yes, yes. And at that point in time, there was only one PCR machine on the entire Duke campus.
It's hard to believe, isn't it?
And now it's so commonplace. But back then there was, I mean, not even the medical school had a PCR machine, not even NC State campus, right? And here I am doing PCR. They had just described PCR. And then John and, of course, Tom, they were on all this fungal amplification, and Redis was connected into that. So I was very fortunate to actually come along at a time where I was able to use some of that for my research. But
it took that to figure out your species.
Yeah. So taxonomically, like, what are these rods? Because they all kind of look the same. It's hard to get them to produce spores, and it's a hard group, taxonomy.
When I was here in Gainesville on the postdoc, Jim Kimbrough and one of his students, CC2, they were working on that project, trying to distinguish some of these.
Oh yeah, I mean, we still are. So we just got a project funded where we're looking at like 50 genomes now. So we're going to just keep looking. You know?
Yeah, no, I think it's interesting for you to kind of reflect on this at the beginning of PCR, and now you're in genomics.
Yeah. So
where's it going?
Yeah, so I guess, I don't know. I think, the sky's the limit, right? And you never really know, right? So you always see kind of these new techniques that are on the horizon, but you have to have good questions too, right? So I'm not really sure what the answer is to that question, but the genome of this fungus is very complex. It has a lot of different nuclear stages, and
Rhizoctonia.
as tractable as some of these other fungi to work with genetically. So we're still trying to figure this out, and why this fungus can do certain things. It has a lot of different abilities to grow in different environments. It can associate with different host plants. It can actually be, sometimes it could be mycorrhizal.
Yeah.
So and in other cases it's actually being parasitized by orchids, right? And so but I still think the fungus is probably getting some benefit out of being associated with the orchid, right? Even though it's been challenging to sort of demonstrate experimentally if they're getting any sort of nutrient-derived benefits. But I'm always told them it was like the orchids are a home for them. They actually can exist inside the roots.
Yeah, how stable is that, with Rhizoctonia and the orchids? Is it one of these successional situations?
Yeah, so typically what happens is they will associate early on with the seed, right? And as the seed starts to germinate, they sort of facilitate the germination of the seed. And the orchid can live for a really long period of time underneath the soil before you even see it. Yeah. So there's this association that's going on for possibly many years under the soil. And then eventually, over time as the plant, at least if it's a photosynthetic orchid, it actually dispenses with the fungus. It gets kind of rid of it. But what's interesting is the fungus usually makes survival structures inside the orchid root. And I was always wondering if that's what it was doing with the orchid, so, but we still don't know much.
Now, years ago I read something about, is it sulfur metabolism that the orchid seed can't do without the fungus? Is that one of the things that happens?
Yeah, I can't remember what it is, and you're exactly right. There is some nutritional benefit for them being associated with it, and it's sort of the reverse, right, of what we think about when plants are given nutrients to the fungus, and the fungus is given back. So it's the reverse direction. Fungus is
You're also now curator in the fungus collection, aren't you?
Yeah, actually that was something that I didn't mention this previously, but one of the best mentors I had was Dr. Larry Grand, both of who you know, and I remember you visited the campus that time and we went out looking for cup fungi, and yeah, I still remember that. My wife still remembers that too. She was asking me about that the other day, because we went down, and every time we go to that spot where you found those fungi, it was right along where the water was.
The urnula.
Yeah, we think about you. We're like, that's where Don found those urnula. So yeah, we I don't know. I was really lucky again. So I go to NC State, and what was happening, and I was mostly teaching plant pathology courses. I was also involved in fungal genetics related courses. And then as time was going on, then Dr. Grand sort of asked me if he would, if I would consider sort of co-teaching with him. So he teaches the graduate course, the Introduction to Mycology course, and then he also taught the undergraduate course, Kingdom of Fungi. So the idea was that over time that I would sort of co-teach with him, and it was the best experience you could ever have, because, oh my God, it was him, and there was also Dr. Chuck Hodges, right? So they would actually, always quiz me about certain fungi, and I learned so much from them. And a lot of people don't get that opportunity. They come in and there's no one there. They got to start up. But fortunately for me, I had them to serve as my mentors for a long time, and eventually I took over those courses. But getting back to your question, was Dr. Grand, as part of the teaching of the course, he established a herbarium there. And so he has a lot of unique specimens that are from the southeastern United States. So we got a couple grants from NSF to actually sort of catalog. This is all before like Mycoporter and all that. It was when we had to like call people on the telephone and try to figure out what they have. Yeah. Not even a cell phone. Calling them on a telephone.
Wire.
Yeah, or it was a wire, or we had to write a letter or whatever, or we had to drive there. You had to drive there and look
Why
at.
don't you talk a little bit about Mycological, because you had one of the grants,
Yeah, so basically.
the money in the grant anyway.
Yeah, right. So basically what happened there is since he had this really unique collection of these fungi, and mostly his collection consisted of wood decay fungi that were mostly found in that particular region, and they were from very unique habitats too. So there were things like from the Nag's Head Woods, which is one of the last remaining, sort of forested areas, near the coast, and there were some other habitats that's not coming to my mind now, but it was the uniqueness of these specimens that they recognized that they should be saved. And he also had a lot of specimens for reports of diseases that were the first to ever be described in either North Carolina or North America. So there was a large collection of those, and we actually recently sent Don's, it's a postdoc.
Yeah, Michael Bradshaw.
So we sent some, we've always, ever since we started that project, we've always sent specimens, but it seemed to increase, right? Because of the ability of the specimens to be uploaded to Mycoportal now. And so, yeah, it's actually more accessible now. So we're getting a lot of more requests. And so, yeah, we actually tried to, incorporate that into more broader, projects that people could use as specimens, whereas they were just largely being used for students. We used the herbarium as a teaching tool for students that wanted to learn about fungi and also to preserve them. And that's the way we teach, right? We teach basically from like, you got to know how to identify this thing, and then we eventually, got to know its ecology. Then you got to be able to store it, you got to be able to preserve it, and right on up to getting the genome. I have a single cell now. So, because in a lot of cases, those specimens are not good for that because the DNA is too degraded. But yeah, so we still use it as a teaching tool. And every year we actually put specimens in there that some student found that was unique. So, yeah, so we're always finding something new and we'll just put it in there. But yeah, he was a great mentor. And we actually had a ceremony for him. Did you come to the ceremony? Mm-hmm. Yeah, we had a ceremony to actually dedicate.
For Larry Graham.
Yeah, name the herbarium after him. Mm-hmm. So it's called the Larry Graham Herbarium, Mycological Herbarium, and it's a defined entity on the campus, which took quite a bit of doing to actually recognize that. And so, yeah, we had a small symposium where we invited people to come and participate. And then, yeah, he was touched by it, and he really didn't want us to do it.
Really?
And so I had to go talk to his family. I said, can you talk to Larry about that? We want to do this for him.
Had to do a cell job.
Yeah, so we had to do a cell job on him, and yeah, so finally he agreed to let us do it. But yeah, he's passed away now, but he was definitely a big influence in my life, so.
Yeah. So, anything else?
No, that's good. I feel like I'm talking too much, but.
No,
that's
the idea. Yeah. Well, thank you, though.
Yeah, people will watch this like, oh my God, he talks all the time.
Yeah.
So what are you doing now that you're not president? Anything?
Well, I'm the past president, as you know, and then as of Tuesday, I guess I'm the past. Mm-hmm. So yeah, 1 foot is in the pasture. That's what I said the past means, right? So 1 foot in pasture. Right, and the next was 2 feet, so that's past. So I'll have 2 feet, and then the gate will close behind me, but I'm sure I'll always be involved in some sort of aspect. I was the treasurer, oh my God, 2010, and I've done a lot of service-related functions. I just love the society, and I love interacting with the students and the members so much. It's a really unique family.
Yeah.
Yep.
Well, thank you. Thank you for all your work.
No, and thank you for taking the time to interview me. I appreciate it. And you guys have been great to me too over your career. So I really appreciate it, particularly Meredith. She's been, a really strong advocate for me. So, well, thank you, Meredith.